talk about these vectors that are used in genetic engineering. Let me take a pen. Okay. So these vectors have some specific characters. Basically, vectors are also DNA because they have to transfer or they have to carry DNA. So they are the vehicles. They are the transport vehicles, you can say, one sort of, which can transport or transfer DNA or gene or whatever DNA of interest or gene of interest that you want to clone and transfer. So these vectors should also have some characteristics, some criteria which must be present in a vector so that they can be used in genetic engineering. So what are the criteria? There are 3-4 criteria that are very important for a vector. The first criteria is this: they must be self-replicative. They must be self-replicative. Means what? The vector DNA should replicate itself without depending on the replication of the chromosomal DNA. So they can increase their number inside by replication. So they must have an origin of replication of their own so that they can replicate. This characteristic is present where? In plasmids, in bacteriophages. So that's why they have been used as a vector. It must have, hello? Are you asking something? It must have one or more unique cloning or cleavage site. Okay, cloning or cleavage site, where that means restriction sites, and that must be unique, means it must be present only once. So if this is a plasmid vector and if there is a restriction site here, say echo R1, it should be present only once. Otherwise, what will happen if it is more than one? Suppose it is also here, then when you digest it with echo R1, then what will happen? It will produce 2 fragments, one from here, another from here. So this will, and you are going to add another fragment, that is insert, that has been obtained from some genomic DNA by echo R1 digestion. So we are going to mix that also with this. This is the insert DNA, say. So when you mix them for cloning purpose, there are 3 fragments, this one, and this one. So how will they ligate? It will be difficult for this vector to ligate this in proper sequence, isn't it? So an restriction endonuclease should be present only once. In that case, what will happen? This this vector will split in a single linear DNA molecule. It was circular, but when it will be nicked here by this echo R1 digestion, it will produce a single DNA strand like this with sticky ends of echo R1 overhangs. So now you are mixing this vector with a insert. So chance of this vector introducing this insert inside it is more. So it is not wise to have more than one echo R1 or BAMH1 or whatever cleavage site that is present. That must be present only once in a vector. In insert, because insert, you are going to obtain the insert from some genomic DNA. So the way the insert is produced is variable. It may be produced by PCR method or it may be produced by restriction digestion like this. So since you are digesting this genomic DNA with echo R1, so it will have many restriction echo R1 sites. This is not a vector. You are going to isolate a fragment from here. So number of restriction site, unique restriction site does not arise here. The number of restriction sites may be more. Okay, but in vectors it must be only once. It must have at least one easily detectable marker. What is that? That means usually that is a antibiotic resistant gene or a LAGZ gene. The gene for beta galactosidase enzyme. Okay, this LAGZ synthesizes beta galactosidase protein. You must have read that in lac operon, isn't it? So why we need to have these markers in the vector? That means this is a vector plasmid DNA. Say this is a plasmid vector. So it has a origin of replication. Say here first characteristic, this one, with the help of which it will replicate. It will increase its copy number. It has several restriction sites. Say here at this place you have introduced several restriction sites together, and say the restriction sites are what are the list is like this: eco R1, BAM H1, SAL1, Hindi 3, KPN1, like that. So 6, 7, 8, 9, 10 different vectors have different combination of these restriction endonucleases. Okay, so when you introduce this is in such a package at a particular place, okay, these restriction endonucleases, you synthesize them and you introduce in the vector, then this particular region is called, this particular DNA is called polylinker. Polylinker, or it is also called MCS, multiple cloning site, because here you have introduced many such linkers. Basically linkers are what? Linkers are small DNA fragments that contain a restriction site. Okay, say if this is a DNA fragment and it contains GAA, TTC, NN, 2-3 more nucleotides. Okay, NN, CTT, AAG, the opposite strand. So this is a linker. Okay, you can synthesize such linkers and you can one after another you can ligate them. This is another linker, this is another linker, this is maybe this is eco R1 linker, this is SAL1 linker, this is BAM H1 linker. Like this, if you attach several linkers together, it makes a polylinker. Okay, and if you introduce this entire polylinker inside a plasmid, then you are introducing a package of restriction endonucleases at a particular position so that cloning can be done at that position. Okay, so these are unique restriction sites, and you have some genes, marker genes. So this one is ampicillin resistant marker gene. Why this is provided? Because when you will clone something here, something at this position in the restriction endonuclease position, wherever the site is there, say you are trying to clone at eco R1 site, so an insert will be inserted here at this position, isn't it? And then what you will do, you will introduce this recombinant DNA into a host cell. That we studied in our first class, that after making the recombinant DNA, that has to be introduced in a host cell. That is basically an E. coli cell, E. coli AMP negative cell, isn't it? AMP sensitive. When this plasmid will get entered into it, it has its own chromosome, but when this plasmid is introduced, it becomes AMP positive, AMP resistant now, and that is why this AMP resistant gene is provided here. It is a marker. So when you will how you will distinguish or screen those cells which have been transformed by this recombinant DNA? By plating the cells in ampicillin containing plates. So when you will plate them in AMP positive plates, agar media, then what will happen? Those cells which have not been transformed, which do not contain this plasmid, okay, only the host cell, that is AMP negative, AMP sensitive cells, if they have not been transformed, because every reaction is not 100%. So many of them will not get transformed, but others will get transformed. Okay, so some of them contain this plasmid and some of them do not contain this plasmid. So when you plate both of them You cannot distinguish bacteria by seeing with your naked eyes. They are present in culture, they are present in culture. So these are the bacteria. Many of them are without plasmid and many of them are with this plasmid. They have been transformed. Okay, so which one you expect will grow here? Those which have been transformed over here by plasmid, isn't it? Because they now contain, the plasmid contains the AMP resistant gene, and hence they can break this ampicillin in the media and they can grow. So these are the colonies that are being formed. All these colonies are those cells which have now become resistant, ampicillin resistant, because of the presence of the plasmid within them after transformation. This plasmid is a recombinant DNA, isn't it? But there are many which are non-recombinant. While this, when you are trying to clone them, there will be result where you will get, there will be many which did not introduce this insert. So there will be non-recombinant plasmid also. So how do we distinguish between these two? Okay, both of them are ampicillin resistant. Okay, the self-ligated one and the recombinant DNA also, both of them are ampicillin resistant. So how do we distinguish them? We will see in the next slide. And that is this LAC-Z gene is about. Okay, or there may be some other antibiotic resistant gene present at this position where you are trying to clone the insert. Either the LAC-Z will be present here or there will be some antibiotic resistant gene. Okay, we will see in the next slide. So these are the three features a plasmid must possess to become, not plasmid, any other DNA molecule which has to qualify as a vector must contain these three features. These are the three criteria that must be present in a vector DNA. Self-replication, unique cloning site, and marker genes for their screening of the recombinant strains. Okay, and this one, the vector must be smaller in size and stable in the host cell. That is another characteristic. That is, you can say that is primary, because if the DNA is not stable in a particular host cell, if the DNA gets degraded because of another restriction, or it gets recombined with the chromosome because of the REC-A positive nature of the host cell, then also, even if it is stable, if it gets recombined with the host chromosome, it will be difficult to identify where your gene of interest has gone. That cell will become infect. It will be a transgenic or modified organism. Okay, it will show antibiotic resistance, as well as it will also get that particular characteristic that you have introduced in that cell. But at the first time, at the beginning, when you are using E. coli host, you are just using this host to keep your recombinant DNA within it. You might be using this recombinant DNA for some other purpose. Okay, you might be using this for cloning in yeast or for cloning in human, not human, I mean human is not, typically being used because of the ethical reasons, but someday there are other methods. CRISPR-Cas9 is being used for that purpose, for gene therapy or something else. But maybe other organisms, plants, etc. Wherever you are trying to do some change, make some change in their gene, or you try to express an extra gene within that organism, you have to transfer that recombinant DNA first in an E. coli cell. This is just a house where you can say it is a temporarily you are storing that plasmid or recombinant DNA molecule within this cell. When you have to transfer that into some other host, you will culture them and culture this bacteria and will isolate your recombinant DNA from here, and you will then go for next round of cloning into that host. You will transfer this into that host. So these are sometimes, these vectors are shuttle vectors. That means they can shuttle between these prokaryotic and in eukaryotic cells. Okay, this kind of vectors which can shuttle between them are called shuttle vectors. That's the basic cloning vectors. These are the basic cloning vectors that are most frequently used. Plasmid vector, Phage vector, Cosmid, Phagemid. We have already talked about them a little bit, and we will see one by one how they are used. And this is the list by which this is the insert size. আবার কেউ একজন একসাথে জয়েন করে না, প্রবলেম ক্রিয়েট করে। হ্যাঁ, এগুলো হলো these are the size of inserts that you can clone in these vectors. In plasmid vectors you can clone about 5 kilobases. This is almost the maximum size. This is the maximum size, 5 kilobases, that you can clone in a plasmid vector. You can go for 6 kb, 7 kb also. I have during when I was working at ISCB, there was in another lab some people were trying to clone this topoisomerase gene, which was quite big, about 6 kb, and they were finding it difficult, and ultimately after 6 months or 7 months of trial they were successful in cloning that, and that was 6 kb. So when the size increases, in my case also, my plasmid, I was trying to make a target vector, gene targeting purpose, and my vector was initially it was a small vector, 3 kb vector, but after several rounds of cloning the size has become about 8 kb or so. So when I was making those modifications, when the size has increased, the things become difficult. Even if you want to clone smaller size fragments, it doesn't happen easily. Okay, so this particular size of a vector is important, probably because the origin of replication that is actually present in the plasmid is on the basis of its size, how long that origin of replication can handle. I mean, what will be the size of the replicon that origin of replication can handle, and if the size of the plasmid is too big, then it will be difficult for one. As you know, in a single plasmid or in a single bacterial chromosome, there is only one origin of replication. So definitely that will have difficulty in replicating the entire chromosome or this recombinant DNA when the size becomes larger than what is expected or what is naturally present in the system. Okay, so whatever the case may be, the 5 kb around is the maximum insert size that you can clone in a plasmid. Less than that you can definitely clone, but more than that will be difficult. In case of phages, you can clone 6 to 23 kilobases. You can go up to 23 kilobases size. So when your insert size is more than 5 kilobases, you will prefer to clone them in phages. Okay, and even if when your size when the size of the insert is more than this, it is about 40 kb, 45 kb, then you prefer to clone them in Cosmids, or recently there are the Phagemids are being used. Those are also one type of hybrid vectors, Cosmids or Phagemids. Okay, so 40 to 45 kilobase insert size can be cloned in Cosmids. I will show you why you need to clone 40-45 kilobases or more. Even these high capacity cloning vectors, you go for these high capacity cloning vectors to clone higher sized DNA. When you require to clone 100 to 300 kilobases, you use bacterial artificial chromosomes, and even when you can go for higher 1.5 mega base pairs, means 1500 kilobases you can clone in each artificial chromosome. So these vectors are required for genomic research. This is also Cosmid is generally used. It used to be used. It was made for this purpose, for doing genomic research or for making genomic DNA libraries. But genomic DNA libraries are mostly made in Phage DNA. Okay, because most of the gene which can be which have a larger size. Most of the genes can be fit within the 6 to 23 kb range. In fact, many genes, if a DNA strand is 23 kilobases long, it will contain more than several genes within that, maybe 3, 4, 5, 6, 7 genes. And if if the sizes of those genes are more, then of course the number will be less. Okay. Usually you will see different types of genes are there. Some are maybe very small, maybe 200 base pair long. One of these such gene is you can generally it is being said that if the protein is about 50 amino acid long, a 50 amino acid long protein can be produced by 150 nucleotides. Means there will be 50 genetic codes required, and each genetic code is comprised of 3 nucleotide, 3 base pairs. So 50 into 3, I mean 150. 150 base pair can be an open reading frame theoretically. So open reading frame that we call a gene is also called open reading frame. So the smaller size can be 150 base pairs. A little more because there will be some upstream and some downstream regions in all genes, and those are for regulation purpose. The RNA polymerase will come and bind to that position, the promoter region, and where the termination will occur, there are some extra nucleotide sequences. As in case of eukaryotic system, I told you that all mRNAs end with a poly A tail, isn't it? So the ORF may be small, the poly A tail may be longer than that in certain cases. There are some Phagemids which are also a combination of Plasmid and Phage. They are made for different purpose, and they can be the insert size can be smaller, 5 kilobases. They are generally made for making single stranded DNA for two purposes. One is for sequencing purpose, because that Sanger's dideoxy method of sequencing, there is a method, Sanger, I told you about that. Sanger was the person who first discovered this process of invented, you should say, the process of DNA sequencing, okay, by an enzymatic method. There are other people like Maxim and Gilbert, two scientists together. They also developed this sequencing method that was a chemical method that didn't become popular because it was a little bit cumbersome, but this this enzymatic method was quite easy and innovative. We will learn that later on. So for that purpose you need single stranded DNA, and these plasmids can be used to make single stranded DNA. How? We will see that. So these are the different types of vectors. High capacity cloning vector, I told you just now that the name is high capacity because they can be used to clone higher sized inserts within them, and they are usually required for genomic research. What is genomic research? The present era, in the last, last couple of decades was for this genomic research. It is going on. It is being flourishing now, and a lot of data is getting accumulated, and you people might find places in research or some other institution or in some job for data mining. A lot of data is being accumulated for this genomic research. From this genomic research, human data as well as other plant data, everything. Suppose there are so many human beings and sequencing of all human beings have not been done, genomic sequence. So but nowadays it has been quite a number of genomic sequences are available in the internet, but when this will go further, when everybody will get sequenced, the day will come when almost everybody will have to sequence their DNA for some other reason, to know what is if there is any genetic defect or like that. Then you will have this everyone, doctors or medical science, will have a lot of information in the web in the form of the genomic DNA sequences, and now the task will be data mining. I mean, here when the coronavirus is, you can understand the one thing, that this coronavirus is effective in certain human beings and it is not so successful in certain other human being population. Why it is like that? If you want to understand, say this Maharashtrian, it is more, I mean, this coronavirus is more compatible with the Maharashtra people than maybe Bihar or some other places, or Odisha, or maybe West Bengal, I don't know. Like few days ago we were talking about why it is so, I mean, European countries are so developed in medical science, America is so developed in medical science, but they are the most sufferers of this coronavirus attack. So why this coronavirus is finding it suitable to infect those people? They have a different origin. They are Caucasians, and we are probably Mongolians. And when you go to south, there may be some, I don't know. They are what are they? Dravidians. But Dravidian is not. I mean, you have to go further back. I don't remember right now. So they have their own origin. They're different. We cannot call us Aryan. Aryan is historical thing. But if you go for human origin, we are probably Mongolian type of. Yeah, we have to check that, and that human classification where from different caste and creed have originated. So does this virus viral compatibility depend on the human genome sequence? Human to human genome sequence is variable, of course. The genes are same, but the intergenic region, or are all genes same? Right now the information is quite small. You cannot say everything from the whatever you have at the present moment. But when you go for data mining, more and more data search you will find, okay, this is the place where the difference is. Because of this coronavirus is finding it more easier to. That is called molecular compatibility. Okay, when a virus jumps from an animal to human. Okay, this has also happened because we call it, there was a bat from where it transferred itself to a pangolin type of animal, and then from pangolin probably it came to human being. So this was an emergence or spillover, we call it spillover, of viruses from animal host to human host, via a certain other animal host. So why this virus chooses that only animal host, and why particular human being is only being chosen, even when there are many other types of human being are exposed to that? So there is a compatibility that is called molecular compatibility, and that compatibility will be revealed more in this genomic era when data mining will go on. So there will be this course. You can go for certain data mining course parallelly when you are doing this botany honors or MSc. You can go for one thing I will suggest, you can easily go for Python. Do you hear me?
Yes, sir.
Yeah, you can go for learning this language, Python.
Coding language.
Yeah, coding language. My suggestion is you should go for learning this Python or R. Better will be Python. Python is comparatively easy. There is another one, R, that is being used more in statistics, biostatistics and statistics, and data mining can also be done with R, and data representation can be done with R. Python can also be very useful, but Python is comparatively easy, and Python is developing very fast, and this is open source and free, so you can easily learn from any other place. So you should go for a short course, maybe an online course from any other places, anywhere you search for, where from you will get some idea. So start with that. Yeah, gradually you learn, and when you qualify BSc, MSc, whatever what you are trying to do, if you are interested, this will be useful always. even if you are a banker or a railway staff or whatever, data handling will always be there in all fields. And a person who knows Python or R will get significance definitely in his or her place of work, wherever she or he is working, isn't it? That person will get significance, and because that can handle, and chances of getting job will also be more in private sector also. Because there are companies that try to make their, statistical data. They try to analyze what is my growth, how it is being declining or increasing. What you already are studying this biostatistics, na? Chi-square test, goodness of fit, mean, mode, median, isn't it? You are doing that. You are doing standard deviation, standard error mean. Just a little bit of more biostatistics will help you to analyze any type of data. You are calling it bio because your data are bio based. Basically it is statistics, isn't it? So same thing will apply to other places. Okay, you are comparing two group of people. Suppose you are comparing two group of students, or maybe a company has made a drug and that drug is being tried upon two populations. Okay, one population was not given that drug and the other population, that was control, and the other population was given that drug. And you as a biostatistician, you are asked to tell them that drug was effective. Maybe that drug is a growth factor and that was tried upon children. So children, the weight of the children at the 0 day and the weight of the children after maybe 6 months after administering that drug, it become little bit more. But where you have not given the drug, they have also got increased in their weight. So is this weight increase and that weight increase without the drug? What is the statistical, is this growth is significant enough that you can say that this has happened because of the drug? So you have to do that via statistics. There are some tests, significance tests. One of the tests is called student's t-test. So you will apply student t-test to solve this problem, and you will get a result by which you can reject or you can accept that, okay, yeah, this is because with drug, the whatever growth you are getting is significant. Or if it is insignificant, you will say that the drug has minor effect, it has not got that effect. Okay, so these things are basically biostatistics, and you can do that with the help of the Excel also. This Microsoft Excel is also very fantastic, but it has limitation, and Python can go further, many different types of things, interpretations. Okay, you learn Python by some other places, and it will be useful in data mining later on. There will be huge task of data mining in future. Okay, let me proceed further. So we were talking about plasmid vectors and PBR322. Suppose this is one example of a plasmid. This is the first vector that was used for genetic engineering. It is called, it is little larger, 4362 base pairs long, and the basic core of the plasmid vector is from P call E1. There is one. Oh, আরে কাজ করছে না। হ্যাঁ, একটা ভেক্টর আছে, প্লাজমিড আছে পি কল E1, P for plasmid. Everywhere you will see there is a small p that represent plasmid, and this is a cholecinogenic plasmid. This plasmid is responsible for cholecinogen production in Escherichia coli, and this is called P col E1. The backbone of this PBR322 or any other, even this PUC vector, PUC, Plasmid, University of California. Plasmid, these two scientists, Bolivar and Rodriguez, and 322 was probably the clone number that he found suitable for this purpose. So this was the first plasmid. Let us understand first what are there in this plasmid. So first of the thing, that origin of replication is there, that must be present for making it self-replicative, and there are some restriction sites, unique cleavage sites like the BAMH1, EcoR1, SAL1, and there are several others I have not drawn. Okay, only few I have drawn over here. Okay, otherwise it will become clumsy. I don't require all of them. Here PBU2 is another. There is another PST1. So 5 restriction endonucleases are present, have been drawn in this diagram. There are several, but these most useful 5 restriction endonucleases have been drawn. Okay, but their positions are important. PST1 is present in AMP resistant gene. This is AMP resistant gene. Okay, yellow one, and this is, what is the color? Red, light red, or like that. This is a tetracycline resistant gene, TET-R. There are 2 resistant markers, so there are 2 selectable markers over here. Okay, how these 2 selectable markers will be used? Let us see that. This marker is just for selecting the cells in which the plasmid has been introduced. This is the host cell with the chromosome, and as I told you that this is, the host cell is always AMP sensitive, AMP negative, AMP sensitive. Okay, so now after the cloning step, means you have taken a small Eppendorf tube, microfuse tube, within which you are doing this reaction. This is the small volume, about 10 to 20 microliter only, within which you have added the vector and the insert, isn't it? Vector and the insert, and you have added ligase enzyme. Okay, ligase will ligate them after overnight incubation at 16 degrees centigrade, so that these sticky ends, those sticky ends are there, overhangs, sticky ends, they will come and they will hybridize, and the ligase will seal this place, so they will become combined like this. I'm just showing one place, one of the restriction area where this ligation is taking place. This place. So this is taking place in this solution, small volume of reaction mixture. Okay. So the next day, what you do, you add some cells within this Eppendorf tube. You add some competent E. coli cells, E. coli host cells. You add them 200 microliter of E. coli cells. Those are transformation competent. In microbiology, we had learned about transformation, transduction, conjugation. In transformation, we learned that not all bacterial cells are competent. Competent means competent to take naked DNA from outside. If they are not, there are some bacteria like the Bacillus subtilis or Neisseria, gonorrhea, or like that, or Streptococcus also. But E. coli is not naturally competent. It is treated with some chemicals, some bivalent ions like calcium ion, or maybe some other methods are there like cadmium, cobalt, etc. treatment are also there, but calcium chloride method is the most common method, by which if you treat these E. coli cells, they become competent. They were non-competent, now they will become competent to take the outside DNA. So you have added this bacterial cell over here, and this reaction mixture contains the recombinant DNA also. So after this reaction, what is expected, what has happened, that the vector DNA, this was the vector DNA, this was the insert. Okay, so you have ligated them, you have trying to ligate them. So what might happen, one thing will happen, that this vector will reseal without the insert. This is one. Within this reaction mixture, this has happened. Okay, say in 80% of cases you will see this to happen, maybe 50% of cases, but rest of the, in certain cases nothing has happened. Three things can happen. None of these plasmids have they have either they are present in a linear manner. No insert has been introduced, but there is another case where this insert, I'm just drawing the insert in a zigzag fashion. Okay, so in that case there are three situations. In one, we call it rather self-ligated vector, self-ligated, okay, or empty vector, phaka. This is recombinant vector. Here the recombination has occurred. This is I mean, you cannot call it anything. Nothing has happened. That means they're present in the mixture. They could not. This vector has to. This insert has to go inside, and that what happens. These are molecules, okay, and these molecules are moving in this small volume in a Brownian motion. Where is my pointer? Here, okay. Sometime I cannot see this. Within this small volume, these molecules are very, very small, okay, and they are moving randomly, and by chance this insert comes in contact with this vector, and then it gets associated with that. It hybridizes, and then ligase enzyme acts upon it, and then it recombines. So the percentage of this recombination will be less. Maybe you can expect 10% of them, of the plasmid, have got recombined. Rest of them are either in the form of this or in the form of this. So all of them are present in this reaction mixture, isn't it? And you have added the cell. After that, what you will do, you will transform. Transformation is done at 41.5 degrees centigrade for 90 seconds. 90-second heat shock is given and immediately chilled. Okay, so you set a water bath at 41.5 degrees centigrade. You take these competent cells and we mixed with the DNA, put it on the water bath, and you see your watch. Nowadays you will see your mobile that 90 seconds is over or not. As soon as 90 seconds is over, you take it out and you put it in the ice bucket, heat chill. In that time, within that time period, the cell has become, because of the heat chill, it becomes porous, and it takes the outside DNA within it. Okay, so chromosomal DNA was there. It transiently become porous, and if it is the way I have drawn, if it is like this, it will break, it will lyse. So there will be some pores created, and the plasmid DNA, recombinant DNA or resealed DNA, whatever, will enter into the host cell. So there are several types of host cells. Three types of cells are present here after this heat shock process. What are those three types of host cells? One is the cell which did not acquire anything. This is a cell within which nothing entered. The chromosome is there, so it is AMP sensitive. Still it is AMP sensitive, and most of this cell will be like this. More than 50% cell will be without non-transformed cells. There will be some percentage in which the only resealed plasmid has entered. So they are AMP resistant but TET sensitive. How? I will tell you. Number 3, these are the original recombinant cells in which the recombinant plasmid has entered. So there are three types of cells after this transformation, and you have to screen this one and to reject these two. How do you will do? In your first plating, when you are plating for the first time in ampicillin media, say this one is your ampicillin media, this will not grow. The first case, this will not grow because it is ampicillin sensitive, but these two will grow. Now you have to do something else to distinguish which one is this and which one is that. How do we do that? That is called insertional inactivation. There is a term, we use this, insertional inactivation. So this is the plasmid, this is the PBr322, the same plasmid. Okay, here is this region is AMP resistant, the same color has been changed, and this is the TET resistant gene, within which there is a SAL1 site Okay, you are using this Sal1 site for cloning, say. Okay, so what you have cloned? You have cloned this blue DNA at this place. So now your blue DNA has inserted inside this area in the Sal1 region. Okay, you digested the insert also with Sal1, the vector also with Sal1, and you have put them together and ligase enzyme, and you have transformed this, as I mentioned here, as explained you here, into an E. coli cell. Okay, so there are three cases that the cells, most of these cells did not get any DNA, so they are ampicillin sensitive. And there are two cases. In one case, this will be resealed DNA, self-ligated vector, and these are the recombinant vectors. Now you are plating all these sample. I mean, all these bacteria that is being transformed. This one. You are after this transformation. I mean, 41 degrees centigrade treatment, 90 seconds, and chilling. You are now plating. All these three types of cells are in a mixed way present in this culture. Okay, so you don't know which one is what, so you have to distinguish them. You cannot just see and with your naked eye and that which bacteria is what. Okay, you can understand bacteria are so small that you can see a turbid mixture. Bacterial cells are present, but now you have to screen them. So at first chance you will plate them in ampicillin. So what will happen, as I told you, that only these two will grow. Those bacteria which did not get transformed will not grow because they are sensitive, so they will die in the ampicillin containing media. So among these colonies, some of the colonies are recombinant and some are self-ligated products. So now what will you do? You will plate them in tetracycline media. You will treat this as a master plate. You treat this as a master plate, and you will do a replica plating. This is called replica plating. So you plate them in a replica. Replica plating is it does what? You put this this plate with the same size which will fit inside an wooden log is there which will fit within this, and that is being coated with a velvet paper or velvet cloth, and that is kept on the desk in an inverted position. And you just like this is an wooden log, and this is the velvet paper or cloth that has been wrapped around this wooden cloth, and you are just inverting this plate on this like this. So what happens? These colonies get stick to this velvet cloth at the same position where they were present without rotating this plate. Simply you will put it softly and will lift it, and then you will touch two empty plates. One of the plate is with tetracycline, another plate is again with ampicillin on this. So what will happen? The colonies that have stick to the velvet will now come in contact with the media, and their positions will also be same. That's why it is called replica plating, because you are just replicating this plate, okay, so that their positions do not change. This time also you put the plates very softly over this log, and these colonies get touched with the media. Even a single cell, if it comes in contact with this, and if the media gets a single cell over there, it will grow and form a colony. So both these plates are put over here and replicated like that. After overnight of growth, what you find that some of the colonies are not growing here in this plate, but they are present in this, because this is same. This was ampicillin plate, master plate was also ampicillin, and this also contains ampicillin. So all of the colonies will grow here in these two daughter plates, but here you are getting some colonies missing. Why? Because you can see that this particular vector, which is a resealed vector, it has tetracycline gene intact. Nothing has been changed here. Tetracycline resistant gene is present here, but here the tetracycline gene has been disrupted by this insert. This insert has has been inserted inside the tetracycline resistant gene. So the tetracycline gene is now inactivated. So this is called insertional inactivation. So the cells that will receive this recombinant DNA will be TET sensitive, isn't it? And those which will receive this one will be TET resistant. So here whatever cells you are getting, these are the self-ligated colonies, self-ligated clones. Those which have not grown here, so how do you get them? Because they have not grown here, they have grown here. So this is also called negative selection. This is one type of selection. This is called negative selection, because you are selecting them which have not grown. Replica plating is used for negative selection, particularly to clone antibiotic sensitive clones, to screen them. So here they are tetracycline sensitive clones which are recombinant basically, and they have become sensitive because of the insertional inactivation. Inactivation of the of the particular gene of interest inside the selectable marker. The selectable marker is now inactivated. So the absence of the colony indicates that there were some that is the position where the colonies that were present were actually recombinant clones that you can take out from here, from this daughter plate, or from replica or master plate also. You can identify them which particular place the colonies have not grown. That's why this replica plating is there. The position must not change, otherwise you cannot locate which particular colony has not grown. That's why they have to be present in the same manner as it was present in the master plate. You got this or not? Tell me here. Are you getting me or not?
Yes, sir.
Everyone?
Yes, sir.
Everyone?
Yes, sir. Yes, sir.
So this is called
Yes, sir.
recombinant selection. Okay. You write in the chat box. You write yes. In the chat box, you write yes. I will see how many people. If there is any problem with anyone, you tell me. Acha. Now this was a vector which was made for first time, for starting when the genetic engineering was just starting. Still it is used in certain places, certain cases, but that has been become a little bit less prevalent, and new vectors were designed after that with smaller size like this one, and new ways of screening were also developed, because here you have to go for replica plating, so it was a little bit cumbersome. One step extra is involved over here. If you see this vector, this is also a plasmid vector. What is the difference? The size is small, origin of replication is there is an ampicillin resistant gene. This will be common to all vectors. Okay, this origin of replication and ampicillin resistant gene, this will be common to all vectors. And almost all vectors from here, after this PBR322, after that the vectors that were developed, almost all of them were on the basis of this LAG-Z gene. Instead of tetracycline resistant gene, now they are using LAG-Z gene. This is also a selectable marker. I will tell you how it is being used. Okay. So now your cloning sites have also been changed a little bit. As I told you that after the genetic engineering were developed, by genetic engineering method, all these restrictions, not all, there are several that can be used. As I told you in the earlier slide, that they were packaged into polylinkers. So Hindi 3, PST1, SAL1, BAMH1, SMA1, EcoR1, here you are getting 1, 2, 3, 4, 5. You might forget the sequence because it is variable company to company, vector to vector, it is different. But the concept is this: there are several such restriction endonucleases that are present one after another. They are linkers. The linkers have been ligated one after another, making a polylinker. And the polylinker has been inserted here in the LAC-Z gene. After this promoter operator region, this polylinker is inserted in this LAC-Z gene. The LAC-Z gene, in presence of this polylinker, is intact. It produces a beta-galactosidase, a complementary factor of beta-galactosidase, and then that beta-galactosidase enzyme is capable of degrading, lactose is its original substrate and inducer, but instead of lactose, here what is used is IPTG and X-gal. These two things are equivalent to lactose. I have a lac operon video lecture. Okay, you go to that video lecture, that is very descriptive, in-depth video lecture, and you concentrate on, watch that video lecture with full concentration. You write something while you are watching. That is where I myself appreciate myself, because that video lecture is with very high content. Okay, so there you will see that IPTG and X-gal, it has been explained over there also, that at the beginning, when the Jacob and Monod were trying to decipher this lac operon, they used this IPTG as an inducer. Okay, and X-gal was used as a substrate. Lactose is inducer as well as substrate. Lactose is the original inducer and substrate, because of which this LAC gene is present, not for IPTG and X-gal, but IPTG and X-gal give you an opportunity to observe what is happening. IPTG, isopropyl thiogalactoside. Okay, where is the pointer? এটা একটু বড় হতে পারে না পয়েন্টারটা, এই যে এই কোথায় গেল, এইখানে আছে, এই যে আইপিটিজি, এটা ইন্ডিউসার, মানে কি ল্যাক অপেরন কিরকম ছিল, এই যে ল্যাক অপেরন প্রমোটার, ইন্ডিউসার, প্রমোটার, অপারেটর, আর ল্যাক অন্য দুটো জিন বাদ দেওয়া হয়েছে, এখানে শুধুমাত্র ল্যাক জেড জিনটা নেওয়া হয়েছে, এই দুটোকে বাদ দেওয়া হয়েছে, ল্যাক জেড এ ওয়াই দরকার নেই, শুধু এইটুকু, কারণ আমার বিটা গ্যালাক্টোসাইডেসটা দরকার। IPTG, if you put IPTG, what will happen? This I produces a repressor. The repressor is a tetramer. So IPTG, in presence of the IPTG, what will happen? This repressor will not, this IPTG will bind with this repressor and will inactivate it. Okay, so this repressor will not bind here in the operator region, and the gene expression will occur. The beta galactosidase enzyme that will be produced from this Z gene will convert this X-gal into a product which is blue in color, in a blue colored product. That is interesting. Okay, so the colonies having blue color means beta galactosidase enzyme is expressed in those colonies, in those bacteria. Beta galactosidase gene is intact. Okay, now you will understand. That means if you clone some gene in this cloning site, what will happen? The LAC-Z gene will get disrupted, will get inactivated, and so they will now not be able to produce beta galactosidase enzyme, and the colonies will be white. That means the recombinant colonies will be white and the self-ligated colonies will be blue. This is called blue-white screening. So without going for further replica plating from this place, you will definitely have added ampicillin within this to remove ampicillin sensitive cells, but you don't need to go for replica plating, because this plate contains IPTG and X-gal that helps you to distinguish the colonies that grow with the self-ligated and the recombinant. They have a different look. The recombinant are white and the non-recombinant, I mean the self-ligated colonies, they are blue. So you pick up the white colonies, those are recombinant colonies, isn't it? So this is called blue-white screening. So this tetracycline, this concept was replaced by this LAC-Z concept, this blue-white screening, and all along most of the vectors, even the Phage vectors, they will also contain this LagZ as a screening, recombinant screening procedure, this blue-white screening method of recombinant clones. So I will probably stop here because the class will become too lengthy. And this is PUC A. This is called PUC vector, plasmid, generated or developed in University of California. The size is about 2.768 kilobases. There are two versions of it. In fact, there are 4 versions of it. These are 2, PUC 8 and PUC 9, and there are 2 another, PUC 18 and PUC 19. I don't know if there may be other also, because you have to see the catalog, because these are sold by some companies. Okay, and there may be some other versions of this also, but most popular is this PUC 8, PUC 9 or PUC 18, PUC 9. Every time you will get, if there is a PUC 8, there will be a PUC 9. If there is a PUC 18, there will be PUC 19. Why? When you are trying to clone this, this polylinker, this arrow is the polylinker. Okay, this arrow is the polylinker. What will happen? It will get cloned in this way, or it will get cloned in reverse way also. Okay, so this is basically important at certain places. Here it is not important, because whatever way they are cloned, their restriction sites, they can be used. So both the vectors, PUC 8 or PUC 9, both are equally useful. PUC 18, PUC 19, both are equally useful. Okay, the thing is this polylinker is being cloned in because you see, this is your vector, and the ends are, suppose you have, again I am using this EcoR one for simplicity. So the ends contain TTAA as an overhang, and your insert also has TTAA on this side and on this side. So this insert may get cloned like this, or this insert may get inverted, and can also be cloned, because both the ends are same, isn't it? Both the ends are same. So this is non-directional cloning. I mean, this cloning has no direction. It can either get cloned in this way, or it can be cloned in a reverse way, because both the ends are similar. Okay, so here it is not so important, but the cloning, if this non-direction cloning is done in case of genes that need to be expressed, that are required for expression, when you are cloning them for expression purpose or for transcription purpose, then the direction must be fixed. Okay, how do we fix the direction? That I will tell you. This is not in your syllabus, but maybe it will come in the story, then I will tell you later on. In the next slides, there were some Phage vectors, Cosmid vector, Phagemid vector, YAG vector. Okay, now you have the building of this vector, the concept of vector. I have already, in these 2 slides only, a lot of things were there.